So moving on to our applications, again, a diagram of our phloem and our phloem tubes. And you can just see, this is the phloem tube that we've been talking about. This is the cytoplasm. There are these big holes. Now remember, plants have these things called plasmodesmata that are holes, in between different cells so they can communicate, share hormones, share resources, and so forth. These sieve tubes are even bigger than these plasmodesmata, and they're really unique to these phloem cells. I'm not aware of any other large holes like that for any other plant cells. I'm sure they exist. And then like we talked about before, since they're stripped down, there's really very little cytoplasm. These companion cells are there to kind of fill in the blanks and make resources. One of those things, I'm sure you can identify this if I didn't label it, as the nucleus. There is no nucleus typically in a phloem cell. So make proteins to make enzymes and all those things that cells need, that needs to come primarily from the companion cell. And sieve is how that's pronounced, and you may have used it you might have one of these in your kitchen. It's a little metal thing with a wire mesh, and you can use it to sift flour. But a sieve is any kind of a filter, basically. Make coffee is basically a sieve. So interconnected, those companion cells and those phloem sieve tubes. The structure related to the function. Stripped down because it's there primarily to move minerals, amino acids, but primarily sugars from the leaves down to the roots, all other parts of the plant. So make sure you draw one of these sieve tube members here. This got the sieve tube plate. These are holes. And you might want to even draw like a companion cell, got a nucleus. And little holes here. Go back, get my... Ooh, I rated a little too much. Holes or a gap in between the cytoplasm of a companion cell and the cytoplasm. I'll try to draw the cytoplasm in here. So reduced cytoplasm. And we'll say water, sugar, amino acids, move down. This way is the direction of the sink. And here we have up here the source, the leaf, 9 times out of 10. Or, like I said, stems can photosynthesize too. So, you might have to identify the xylem and the phloem. Well, for the exam, you will. So this is a dicot stem, and you'll see there's these they're called vascular bundles here. Or our vascular system, arteries and veins. Vascular bundles. I should get some slides so we can see this. The inside, the little area here, this is the xylem. And this area out here is the phloem. And the way I remember this is that if you think about, you've probably seen images of maple leaves being tapped with those taps, so the sugar will come out. Well, it doesn't have to go in very far, because as you can see, the phloem tubes, and remember that's the sugar, is on the outside, and the xylem is on the inside. So if you just kind of go around xylem, phloem, xylem, phloem, xylem, and phloem. And what I need you to do, hold on, just a minute. Here's the root of a dicot. The very middle, this part here, is the xylem. And sometimes it'll look like a T. It'll kind of look like this. This is kind of like a kind of a Y. The tissue around here is the phloem. So if I kind of maybe choose a color, this stuff here is the phloem. And this middle, these middle parts, it's hard to see there. That's the xylem. Alright. Now, what I need you to do is here, I need you to draw examples of both of those with your vascular bundles. And then, you know, you'll have these two kind of parts. Maybe get some shading, get different colors. There we go. That's it. And then you can kind of make your own key. Then I would have my red be the xylem. So take your time, make it nice, don't speed through it like mine. And then you want the root cross section. Draw it real nice. We know that that kind of Y-shaped structure is the xylem. The cells around it is the phloem. And one thing I want to mention, you will have, there are monocots. Those are different. There's a lot of differences between monocots and dicots. And one of these things is when you cut open their stems and their roots, they look slightly different. They still have xylem and phloem, but in general, you'll see more, it looks like more organization. For example, in the stems, you see these vascular bundles kind of spread out all over the place. No real pattern, while dicot stems form a ring near kind of the outside of the structure. The root, you'll see this is typical of a dicot root and then monocot root. So just be aware of that. Why don't you go ahead and draw those also? So here's another picture I want you to have, just showing you the difference. Dicot and monocot roots. Now the last one is kind of a sick experiment. You maybe are aware of aphids. Aphids are very common. They feed by sticking this big needle-like structure called a stylet into the plant, and they want to tap that phloem, get into that phloem, so they can suck that sugar out and the amino acids and the minerals, all that good stuff, and that's their food. So you can see it's not an easy job. They gotta go through the epidermis, then the mesophyll, then through a companion cell probably, and then they will reach the phloem. And here's kind of a picture. Look how far it needs to go to tap into that phloem. Now what they did, this experiment, they had these aphids put their stylets into the plant, and then they use these scissors and they cut off the stylet so they can just see the sugar oozing. Sugar oozes out, and they can take that sugar and they can study it, because as we talked about, this plant is grown in a chamber with radioactive carbon dioxide, radioactive CO2 that uses carbon-14, and that can be detected by instruments. So we have the leaves, remember, inside this chamber, and you basically have 4 colonies of aphids, and I'm not sure why they don't have the starter colony there. Maybe they left the aphids there and did not cut their stylet off. I'm not sure. But this colony one is about 20 centimeters away. colony number 2 is 40 centimeters, and then the third one is 60 centimeters. And what they did is they measured, and they started the experiment, started to expose the plant to radioactive carbon dioxide. How long did it take for radioactively labeled carbon dioxide to be detected in that area? So what you see down here, just 20 centimeters away, it took almost hour and a half, and then it took about a little over 2.5 hours for it to, so 1.5 hours to get here, and then about 2.6 hours to get there, and then 3.8 hours to get. Oh, it's already, so that's already 1.25 hours. I'm sorry, I get confused sometimes. Our time system is based on 60, right? It's a base 60. So this is base 100 or base 10. These numbers here. So 1.25, that'd be 1 hour and 15 minutes, right? 1.5 hours, 2.4 hours, all this, and 3.8 hours. So as you might imagine, it takes longer for the sucrose to be transported, but this is getting a good idea of how long does it take plants to do this. And you see they calculated the rate, and it looks like the rate was about the same, about 16 centimeters an hour. Is that what you would expect? So good, that finishes 9.2.